For the second part of the video, we're going to continue with the idea now of coupling. So this is part two. And before we dive into coupling, I just want to go back to where we started and think about diseases and variants and just bring up these three words. So Mendelian inheritance, and then the idea of a monogenic or polygenic disease. So you guys already know these words very well. The idea, like, is there one very strong, usually rare variant that can cause a disease, which is Mendelian or monogenic? Or do you need a lot of weak variants or weak things that you break that together those make the disease or make the phenotype? And of course, technique like GWAS are very good at finding monogenic or Mendelianly inherited diseases, right? If many, many people have all the same thing or a very small set of things that can cause the disease, variants that can cause the disease, a GWAS can find those pretty effectively. But if a disease is caused by lots of different combinations of variants, then GWAS is not going to be able to find those variants. And as we've gained more and more knowledge about disease, the scale is tipping from what we thought would be a lot of Mendelian or monogenic diseases to most diseases, especially most common diseases, being polygenic. And this makes a lot of sense when you go back and think about it, right? Because biology is really complex. We already kind of know now that everything affects everything else. So make a change in one gene and it affects many pathways in the cell, in the tissue, and in the body. And so we know that to build something, you need a lot of different genes, and to break something, you need a lot of different genes involved. We often use a fancy word like epistasis to talk about the interaction of things. But really, it's that biology, there's always lots of different things interacting, and the simple cases is the rare ones, right? Things that are monogenic are quite rare. They're the exception. The rule is that you need a lot of things interacting. And so the reason that I care about that is that, well, anyway, that's why you should care about things like coupling, because the majority of what you guys want to study is probably polygenic. And it's not enough to look at one gene or even one site in a gene. So coupling is trying to figure out how do things interact. And it's doing it in a very specific way that's part of comparative genomics and very closely related to conservation. So here's a plot that shows the conservation of different amino acids across the first 24 amino acids of a particular protein. And you guys have seen plots like this before. A bigger letter means that it's very highly conserved, and a set of smaller letters, like in column 8 here, means that there's very little conservation, and you see lots of different amino acids at that position. So it's true that a highly conserved position like that big L at 10 probably means that it's important to have an L there, and that a variant at that position is probably going to hurt the protein. But is the converse true? In other words, at position 4, where we have not very much conservation, right? There's a lot of different sequences that are here. Can we just put anything we want there? And since we're talking about it, you know that obviously that's not true. So the converse is not true. Just because a position is not conserved doesn't mean it's not super important. So here's a quick multi-sequence alignment. There's 8 sequences here, and I'm showing you the first 20 or so nucleotides. And what you can see is there's a lot of sequence variation in this region, and I'm showing the conservation score along the top. And so here you go, in the 4th column, you can see it's all C, so that's really highly conserved. There's an A here, super highly conserved. But then other regions like here, there's like pretty much no conservation. And then there's other regions that are kind of weakly conserved. So the regions that have identities, or the columns that have identities, those are there's nothing interesting going on there. They're probably important. You probably don't want to change them, but we can't learn a whole lot from those. But what about these two columns? take a second and just see, without my help, pause it maybe. Is there anything interesting that you notice about these two columns? Hopefully what you identified is that when this first column that I marked has a C, the second column has a T. And when the first column has an A, the second column has a G. Now the letters don't matter at all. But what matters is that there's some coevolution going on between this column and this column, such that when this column changes, this one also changes. So they're co-evolving or co-varying or correlated with each other. They're not invariant. They're not like this C or this A, that they're always the same. They're slightly conserved, but interestingly, when one changes, the other does as well. That's coupling, right? So those two columns are coupled to each other. We just know that this correlation occurs. We don't necessarily know what it means, but it probably can teach us about how the protein works. So we could do this. We could assign this a score. We could do some kind of a correlation between one column and another pretty easily. And we could do every column to every other column. And that would give us like a covariation matrix. And here's a covariation matrix that I made for a protein that I'm interested in, SARM1, or that my lab works on actually. And what you can see is it's really cool. These bright blue regions are regions that are covarying, like have a high coevolution score. We won't worry too much about how you calculate it, but basically these two would have a very high score, and then like, you know, this column and this column would have a low score. And so the bright blue regions that have a high score identify what turns out to be known domains. So this is a protein domain, this is another domain, and this is a third domain up here. And so that's kind of cool. So you can actually see the protein domains in this protein, right, inside this protein, or gene in this case, just by looking at these coevolutionary signals, right? Which sequences are correlated with each other across different species. The other thing that's kind of cool is you can see that these first two domains appear to interact in some way, right? Because there's like a dark blue here. So these two interact with each other, whereas this one and this one, there's not as much signal here. But what's really cool is that you can see where the second domain specifically interacts with the third domain, and you can see the site, right? So it's along this part of the domain and this part of this domain where these two interact. You can see that there's a weak coevolutionary signal between the two. I shouldn't say we know that that's where they interact. That's where we hypothesize that they interact. There's also some other cool, like little subtle things, like there's regions that there's no coupling or less coupling within these domains. So stronger interactions and weaker interactions, places where this domain might move around. And so this has nothing to do with structure or nothing to do with a known function, but these maps can give you lots of hypotheses about how a domain or a protein actually works. And so the first big takeaway is that low conservation does not mean unimportant. And in fact, when you have lower conservation, you can ask these questions about how are different parts of a protein interacting with each other, or at least how are they associated through these coevolutionary signals. So most importantly, I want you to be able to intuit that, like what do I mean by coupling? If you had to calculate it on your own with a non-formal method, could you do it? But of course, there's a formal way to do this, and it's called SCA, or Statistical Coupling Analysis. And it's pretty much exactly that. So here's from their paper. This is from the Raghunathan lab, which has put out some really cool papers lately that you should check out. So here's a conservation plot of some domain. And within this domain, right, it is itself a domain, and we're looking inside of it. It happens to be a kinase domain. You can see there's some regions that are more conserved and others that are less conserved. Don't worry about the structure for a second. But here, across that whole domain, from position 5 to 165, here's that coevolution plot. So brighter colors here means that things are co-evolving. So that's great, but within that, you can't see a whole lot of structure in this form. But what they realize is that, we don't really care what the position is. We want to just find out what these interacting regions are. So you can apply now any way to decompose this matrix. In this figure, they used hierarchical clustering, and you can see that, oh, look at this is really cool. There's actually these sectors that pop out. Some of them, kind of just interact with themselves and are independent. Other ones are, there's clear sites where they interact with the other sectors. But so this is really cool. So we've gone from a domain down to the pieces of the domain that are independently co-evolving with each other, which implies that they're important. So this hierarchical clustering was just to show a visual example. They actually use something more closer to principal component analysis to pull apart what the different sectors are inside of this, right? To basically like this versus this versus this. And this overall technique, they actually got it from the financial industry because the also really good, they're not bioinformaticists, but informaticists that try to interpret, stock data and buying data, stuff that I don't understand very well, but there's really smart people that work in that industry as well. And so this is actually cool that the biologists are actually using some stuff from the financial world. Sometimes it's the other way around, that the financial people use algorithms that are come from biology. So once you identify these sectors, so here's the steps, right? So you identify evolutionary covariation or coevolution in a matrix. You decompose the covariation matrix, in this case with something like principal component analysis. And then that can tell you set some thresholds, and that can tell you the sectors that you're interested in. They use the term sector here because these are not they may be domains, but they're not necessarily domains. They're usually very different things. So here's this kinase domain. This is really cool. So the 3 sectors that they found are completely new. They're not related to secondary structure. They're not related to known domains. They're not related to known parts of the sequence. There's something brand new that came out of looking across multiple species, so coevolution. And what they found was that there's 3 main sectors that identified almost every amino acid fell into. The sector, I don't think I said it carefully, but from a single pair, right, which is the coupling that we talked about above, you can go beyond that pair and ask like, okay, there's a whole network of co-evolving columns, and you put those together into a sector, right? Basically like one block here. So they call them red, green, and blue sectors. And it turns out that mutations in each of these tended to have to do with catalysis, substrate specificity, or regulation. And they went and did additional experiments and confirmed that, yeah, any mutation in a red sector affected the catalysis. Any mutation here affected substrate specificity, etc. So this is really cool. You guys can now take your gene of interest, run this Scott analysis, and find near a region that you're interested in, that you've been building variants for, that is part of some coevolutionarily conserved sector, and you can try to start figuring out what that sector does. And therefore be able to predict what other variants in other places would do. So rather than just going blindly into a saturation mutagenesis, you now would have a good idea. The last thing that I want to say about this is that this turns out to go even beyond identifying these sectors. And so since this was a kinase domain, they did a little bit more with it. And so they measured for the kinase domain, and here's the wild type, the melting temperature and the enzyme kinetics. So the melting temperature, right, the wild type is very stable, so it has a high melting temperature. And as you would go down to this axis, you'd be losing stability. It's gonna melt easier. And then the wild type has very good enzyme kinetics, and then they get worse as you go in this direction. So they put in a mutation of the blue sector, and they got this result. So the blue sector they expected to have something to do with, this is a different paper, I think, than the one before. But they expected it to affect the melting temperature, and indeed it did, without having any effect on the enzyme kinetics. Whereas this mutation affected primarily the melting temperature, but it also had a small effect on the kinetics. So they took these measurements. Okay, and then they also did a third mutation, G216, that was in their red sector, which they predicted would only have an effect on the catalytic ability, and that was the case. So you can see that it has pretty much no effect on the melting temperature, but a pretty extreme effect on the enzyme kinetics. So, okay, no big deal. They predicted that, but you still had to go and do an experiment to know how much does it going to affect. And there's other figures where they have a bunch of different mutants and they all fall into different planes, as you'd expect. But this experiment's really cool because now they said, well, what if we do a double mutant? Well, we predict that if we add this to this, that you'd get this white circle here. And then they made the mutant, and this is what they saw. And similarly, they made this double mutant. Here's what you'd predict, and here's what you saw. So, again, if you guys take your gene of interest and you have a few mutations in these sectors, you can actually now use them to predict what other mutants and what multi-point mutants, what kind of phenotype they would give you. And you can go the other way and say, okay, I want this specific phenotype. I want this to be the melting temperature. I want this to be, and use these kind of data now to reconstruct the protein so it works the way that you want. And another paper has designed a tool to try to do just that called Prot. And here they use, rather than using energy-based mutants, so there's many algorithms that try to predict folding and things like that for stability, where they're actually using these evolution-based mutants, which is SCA, to try to figure out how they can add the right mutations to stabilize any protein that you want or any protein variant that you want. So you guys can basically design different versions of your protein using these coevolution-based and coupling metrics. So that is it for this second part of the talk. Make sure you think about coupling, try to draw it out or do something with it. And definitely play with the Raghunathan paper and this Prot site.